Alright, now we're going to look at how to set up a monohybrid cross. In this sample problem, we have a cross between a brown-eyed male and a blue-eyed female. Whenever you're given a problem, it's a good idea to go ahead and highlight the important information. Brown-eyed male, blue-eyed female. We're also given further information that brown eye color is dominant over blue, and that the male genotype in this case is big E, female, little e. Once I've picked out the important information in the problem, I can then go about solving the problem. The first step in solving a problem is to go ahead and set up my key. In this case, I'm talking about eye color, given that brown eyes are dominant over blue eyes. I'm going to choose a letter that's really clear to tell the difference between capital and lowercase. We're talking about eye color here, so let's use E's, where capital E will signify the dominant trait, which in this case is brown eyes. And little e signifies the recessive trait, in this case, blue eye color. I now have established my allele key. Next, I want to go ahead and figure out what my parent generation, the phenotypes, are. The phenotype, what they physically look like, brown eyes and blue eyes. Next, I now want to fill in my genotypes. In this problem, it's given to me that both of these parents are homozygous, meaning they have the same version of the allele. The male is homozygous brown, which would make him big E, and the female is homozygous blue, little e. It's important to note that the only way to be recessive, to show the recessive phenotype, is to be homozygous for the recessive allele. The next step is to go ahead and look at what gametes I'm going to have. Let's look at this in a little bit more detail for just a moment. Let's take the father, who has the genotype big E. These are two copies, 1, 2, of the E gene found on corresponding homologous chromosomes. So just in a simplified view, I'd have my big E. Let's think about what happens to these chromosomes during meiosis. During meiosis 1, the homologous chromosomes segregate, that is, that they separate from one another, moving in opposite directions to opposite ends of the pole. So that if I end up with two cells, 1, 2, one of my homologous chromosomes would go into one cell, the other goes into the second cell. So that here I would end up with big E, and over here I'd also end up with the big E. Okay. Then in meiosis 2, the sister chromatids separate. This happens to two cells. Okay. So that I have a single unreplicated chromosome, each having the E. Now, these are now eggs, these are now sperm. These are the gametes that have been produced. This is what I want to fill in next. First, recall, I have my parent phenotype. Then I have the parent genotype. Now I'm going to figure out what the gametes are. The father can give a big E or a big E after going through the process of meiosis. The mother can give a little e or a little e. Now that I have my problem all set up, it's time to solve the cross. In order to do this, take the gametes from one parent and put them along the side of my box. So I have two gametes here, so I'm going to have two rows. I'm going to take the other parent's gametes and put them across the top of my boxes. I have two different gametes listed here, so I'm going to have two different columns. Now I just need to go in and fill in the information. Convention tells us that the capital letter always comes first. There we go. Now I have my F1s. Inside the box here, These children are my F1 generation. So what do they look like? Well, we know that brown eye color is dominant over blue, and in order to show that dominant phenotype, I only need one copy of the brown allele. In this case, I have 100% heterozygous brown eyes. Heterozygous because all of my children here, 100% of them, are big E, little e, having two different alleles. If you follow these basic steps every time you approach a problem, you'll keep yourself straight and you'll answer the problem and the question that's being asked. First, highlight the important information. Second, set up your allele key. Third, list the parent phenotypes. Fourth, list the parent genotypes. Fifth, write out the gametes. Sixth, do your cross. 7, answer the question being asked.